So fNIRS and EEG are two technologies that people have applied to measure the state of what's going on in people's heads. And so fNIRS stands for functional near-infrared spectroscopy. In EEG stands for electroencephalogram. It's a bit of a tongue twister, that word. They're measuring two very different types of things. fNIRS is the optical measurement, so it's measuring the interactions of um, the molecules and the light that you pass through it, near infrared light. And EEG is measuring the state of the electrical activity of your nerves. So a typical EEG setup is a sort of a whole head device with lots of different electrode points, and it's measuring the electrical activity between different zones on your head. So the electrical activity is generated by your neurons, and that's how it generates a signal. And similarly, if this signal, it has to be compared to lots of different points in the hat that you're wearing and processed quite intensely to get clean data from. fNIRS is comparatively a lot more simpler. The near infrared light again passes into the brain, is either absorbed by blood or reflected. And we use two different wavelengths, which correspond to the oxygenated blood and deoxygenated blood because they absorb light differently. Um, and from that, we can over time look at how the overall vasculature is changing. So when your body is doing work, it requires basic things like oxygen and nutrients, and that is delivered to all your organs, and your brain is no exception, by your blood system, your vasculature. When your brain engages in work, there's a cascade of signals that tell it to recruit more blood to that zone. This is called the hemodynamic response, and our technology can measure that happening in real time. And there are aspects of the hemodynamic response which we can use to infer the types of activity or the types of attention that are occurring at the time of the measurement. Um, because it is a sort of a more global thing, it has the compromises of one, we're looking at only the electrical activity, and your brain is not just made out of neurons making this electrical activity. Your brain is supported by an entire network of cells, which again they help provide the support structure. So astrocytes are one of the more populous type of cell, and their main job is to provide support and food for the neurons that are doing the sort of thinking work, as we would probably consider it, or the controlling your muscles type work. And they also require nutrients, but they're not producing the same electrical signals that EEG would do. So they're comparatively different technologies in that they're measuring different things, and they have their own sort of trade-offs. fNIRS, because it's simply a lot less invasive. You don't have to have lots of different electrodes. It means we can package this into a sort of smaller thing like a baseball cap, and you can go around your life without it affecting it too much. And it also gives you insight into other, information you may also be interested in, like your heart rate. So that I would say those are the main differences between fNIRS technologies and EEG technologies.